Webinar today on the evaluation of TCR-pMHC Affinity and its implications for T-cell therapies. At Immudex, we've been collaborating with Quality Assistance for some time now, and earlier this year, we have announced a partnership on TCR affinity measurement services, where Immudex is providing the TCRs and peptide MHCs, and Quality Assistance is using these reagents to measure TCR-peptide MHC affinity. So today, we're very pleased to present you this joint webinar with Quality Assistance to show you the results of this partnership. So before we get started, I'd like to introduce our speakers for today. We're pleased to welcome Dr. Fabian van der Meers. Fabian has many years experience in the field. He's done several postdoctoral positions after his PhD. Some of these are at the University of Pennsylvania in Philadelphia, also in Australia in Perth, and at the University of Liège in Belgium. So he's worked on different topics related to the use of immunotherapies to fight lung cancer and mesothelioma. Since 2013, he's joined as an R&D scientist at Quality Assistance and has been involved in the development of technologies such as flow cytometry, qPCR, immunoassays, cell-based assays, and surface plasmon resonance, or SPR. And since 2016, Fabian has been the R&D technical leader responsible for the implementation of new platforms, including Biolayer Interferometry, or BLI, digital PCR, and NGS. I'd also like to introduce my colleague from Immudex, Lin Zhu. Lin has worked in immunology for many years, especially focusing on the detection of antigen-specific T cells. She's joined Immudex as an R&D scientist in our custom solutions and services team at Immudex, where she was responsible for producing both peptide MHC and TCR monomers, as well as using these reagents to detect and identify antigen-specific T cells. The agenda for today is, firstly, Lin will give us an introduction to Immudex's TCR solutions and the workflow of TCR manufacturing and QC. And then we will hand over to Fabian. He will talk us through the importance of TCR affinity measurement in the context of developing T-cell therapies and give us an introduction to both SPR and BLI. And then he will take us through the results of a case study looking at NY-ESO I as a model TCR. So finally, Fabian will wrap up by showing us some of the best practices for TCR-peptide MHC affinity measurement. So without further ado, I will hand over to Lin to get us started with the presentation.
Thank you, Hazel. Hi, everyone. I'm very happy to have the opportunity to introduce our TCR solutions in Immudex and present the workflow of TCR manufacturing and how we QC the produced TCR molecules. For those of you who are not familiar with Immudex, we are a Danish-based biotech company specialized in providing antigen-specific immune monitoring reagents, such as our MHC dextramer technology. All the products from Immudex are developed and produced in Denmark. We have also a dedicated American sales and a custom support team based in the States, who are always willing to help with you with your projects. As you know, we have more than 10 years experience in manufacturing MHC-like proteins. With this, we have recently developed the TCR solutions. More than 25 TCRs so far have been successfully produced, include TCRs recognizing complexes such as MHC class 1, MR1, and the non-classical HLA-A. We have rigorous QC procedures to control the quality of the produced TCR molecules, including functionality tests. I will talk more about it later. So, what is so special about the soluble TCR made by Immudex? Well, as we know that a full-length TCR contains variable and constant domains, as well as transmembrane and cytoplasmic domains. Without the anchoring transmembrane domains, it is quite a challenge to produce a stable and soluble TCR monomer. We have optimized our TCR production platform, which allows us to stabilize and purify TCRs to bypass these limitations for advanced immune monitoring as you want. We provide high purity TCR monomers in different amounts, and if you are interested in detecting the peptide MHC complex presented by the antigen presenting cells, you can also purchase TCR dextramers with 4 available fluorochromes. A TCR manufacturing project normally consists of 3 phases. trial expression of TCR chains to see if we can express the alpha and beta chains in our platform. If the answer is yes, we will then move on to phase 2, which is a phase that the TCR is produced and QC'd. Once TCR passes all the Immudex QC criteria, we will then enter phase 3, deliver the product. Let's now take a closer look at the 3 phases. Phase 1. Next, please. It normally starts with receiving the individual sequence of the alpha and beta chain of the desired TCR from you. We will then design expression contracts for this and go through an initial test expression phase. As I said, just to make sure that we can actually make those individual bits. The reason we do this is for some TCRs, the sequences simply cannot be expressed in the system we have in-house so far. If things look promising, we can express both chains as shown in this gel, indicated by the arrows, the orange for the alpha chain and the blue for the beta chain. We will then move on to phase 2. In phase 2, we will start scaling up and produce larger quantities of both the alpha and beta chains separately and purify them. After purification, we will refill the TCRs by mixing those 2 components together to get what we call crude TCR monomer. We will then purify this crude TCR monomer 2-dimensionally and check that it actually comes out with the right composition, with the right size. This is also the first round QC we run on the purified TCR by using the gel to determine the protein size. They have the right stoichiometry and the purity. The second round QC is to test the bioaccumulation degree of the bioaccumulated TCR monomers. We call this a shift assay. It is also gel-based. By using this shift assay, we can not only determine the bioaccumulation efficiency, but also double-check the protein size and purity. And if everything looks fine, we will then run the third QC. We will take the bioaccumulated TCR, attach it to our dextran, as you can see in the black backbone. And perform a specificity and functionality QC assay, which is developed by Immudex in-house. It is a beads-based assay to mimic the TCR binding of its cognate antigen. As you can see here, we can use this TCR dextramer to bind the beads that are coded with either the cognate peptide MHC complex, which is the light blue one, or this unrelated or negative peptide MHC complex. If the TCR dextramer can recognize the beads with its cognate pMHC specifically, for example, here giving this pretty pink positive signal, but not the negative or unrelated peptide pMHC complex as the sign and the gray peak showed here. If this is the case, we will then consider this TCR is properly refolded and functional. With this, we can then move on to the product deliverable phase 3. Once you receive your TCR monomers or TCR dextramer reagents, you can probably use it for cross-reactivity screen, TCR-like antibody applications, quantification of antigen-presenting cells, cell therapy, analysis of TCR-pMHC complex affinity, which we will hear a lot more about it today from our partner quality assistants presented by Dr. Fabian. On top of this mentioned applications, TCR solutions can also be used for validation and characterization of candidate TCRs or quality control of peptide or cell-based vaccines. With this, I would like to pass the microphone to Dr. Fabian, who will tell us the story of evaluation of TCR-pMHC affinity. Thank you.
Thank you, Lynn, and hello everybody, and thank you for joining this webinar dedicated to the evaluation of the affinity between the TCR and the MHC complex and its implication for T-cell therapies. So let's start first with a short introduction about the company. So Quality Assistance is a contract research organization, and we provide to the pharmaceutical industry analytical services. So we can develop, validate analytical method in order to produce QC testing, stability studies of different kind of product in order to prove the quality, the safety, and the efficacy of the medicines. We are working in the GXP environment in compliance with the EMA and FDA regulations, and the company was created more than 40 years ago. Now we are more than 250 employees working on a single site in Belgium. And in terms of product, we are mainly working on innovative human product, such as biotherapeutics, new chemical entities, mRNA, viral vectors, vaccines, nanomedicines. And of course, the goal of this webinar today will be to talk a little bit more about the cell-based medicinal product and especially TCR. So TCR is the T-cell receptors. It's a receptors located here in the surface of T lymphocytes. And the TCR is composed of a highly variable alpha and beta chain. The primary function of the TCR is to recognize the antigens, which is presented by the major histocompatibility complex, or MHC. And this MHC is composed of a MHC class 1 chain and the beta-2 microglobulins. This MHC is located in the surface of antigen-presenting cells. And when the TCR bind to the MHC complex, this will provide an intracellular activation signals to the T cell, which will activate the T cell. This T cell will be able to produce cytokines and will get an activation signal and cytotoxicity activities. This interaction between the TCR and the MHC is very crucial for the adaptive immune system development and also to protect against pathogens and cancer cells. In most of the case, the interaction between TCR and MHC complex is of low affinity. So most of the time it's in the micromolar range. But for therapeutic application, different companies try to modify the affinity of the TCR to get higher affinity TCR. These high affinity TCR are currently evaluated in different clinical trials, mainly for cancer research. And the main targets are the NY-ESO I, MHA3, MHA4, or MARCH1. Very recently, it was in August 2024, the FDA has approved the first TCR-based cell therapy. So it was the TCR MHA4 TCR from the company Adaptimmune. In terms of regulation, there is no specific guidelines for the moment for this kind of product. So we have not the information of all the tests needed to approve this kind of product. But of course, the affinity between the TCR and the MHC will be required in a moment during the development of your product. This because this has a strong impact of the efficacy of the treatment. Because if the TCR has a too low affinity, this will create an inefficacy of the TCR, as it will be not possible to interact with the MHC complex, and also give potential cross-reactivity. If the affinity is now too high, this will create an overactivation of the T cell and potential side effects. So it's really a balance to find between low and high affinity to improve the TCR and optimize the efficacy of the treatment. So this modification of TCR are performed by molecular engineering. So now let's talk a little bit more about the affinity and the kinetics. So what is the affinity and kinetics and how they are determined? So if we look at this interaction, here we have two different molecules, A and B, that interact together to create a complex AB. And this is characterized by an association rate, which is the creation of the complex over the time, and which is dependent of the individual concentration of the molecules, but also on the Ka constant, which is the association rate constant. In the other part, we have the dissociation rate, which is the disappearance of the complex over the time, and which is dependent of the concentration of the complex, and the Kd constant, which is the dissociation rate constant. These two constants, Ka and Kd, are the kinetics constant, and they characterize the speed of the interaction. So how fast is the interaction between molecule A and B. At the equilibrium, this association rate and dissociation rate are equivalent, and we can measure a new constant, which is the ratio between the kinetics constant. It is the affinity constant, so the Kd value here, which is expressed in concentration unit. So now, how can we measure the constant, so the kinetics and the affinity constant? So there are different technology, but at Quality Assistance, we mainly use two different technology. The first one is the surface plasmon resonance, or SPR. And we use the BioCore T200 from Citiva to do this kind of experiment. So the principle of this technology is to first immobilize the first molecule of the interaction on the sensor chip. So this is a glass support on which we coat a thin gold layer. And there are different kind of sensor chip, and based on the chemistry of the sensor chip, we can attach the first interactant, so here, which is the ligand. And the second molecules is injected in the microfluidics of the equipment as a dilution series in a continuous flow. This analyte will reach the flow cells where the interaction occurs between the ligand and the analyte. In terms of detection, a light is sent to the sensor chip, and this light will be reflected to the detectors, and this reflection will depend on the nature of the molecules. So a part of this light will be absorbed by the surface and will create a decrease of the signal in the detector for a specific angle called the resonance angle. And based on what is attached on your sensor chip here, this reflection angle will be completely different. So in SPR, we will follow the evaluation of this resonance angle with the time. So here is an example. So in the first situation, we just inject buffer, and we obtain a signal which corresponds to the baseline here. In the second situation, we start to inject the sample. This sample will reach the flow cell, start to interact with the immobilized ligand, and we create an increase of the signal. We can reach a plateau at the equilibrium. And this first step is the association phase, and will allow the determination of the association rate constant, the Ka value. After the injection, we start to re-inject the buffer, and we'll start to dissociate the complex. So we observe a decrease of the signal corresponding to the dissociation phase and the determination of the Kd value. At the end of the dissociation, most of the times we are not able to come back to the baseline. So before injecting a second sample, we need to use a regeneration step with a regeneration solution to remove a residual analyte solution. Based on this sensorgram, we are able to determine the different constant based on a mathematical model which is applied to the experimental data. Most of the time, we use a 1-1 fitting model. The second technology is the Biolayer interferometry, so it's BLI, and we use the octet rate system from Sartorius. In terms of principles, it's quite similar to the SPRs, but we use here biosensors. So these biosensors are composed of two different surfaces. A first internal surface, which is common for all the biosensors. The second one, which is created by what is attached on the tip of the biosensors. So here in the first situation, we just bind the ligand. And in the second situation, we have the ligand and the interaction with the analyte. So this will modify the layer here in the tip of the biosensor. And this will modify the reflection pattern when the light is sent in the biosensor. So this pattern is modified between these two solutions with a shift called a wavelength shift. And in BLI, we can follow this wavelength shift expressed in nanometers over the time to create this sensorgram and determine the kinetics and affinity constant. So these two technologies are able to determine the affinity and the kinetics, but of course have some difference. The main difference between SPR and BLI is the fluidics environment. So in SPR, the reaction occurs in a fluidic environment, but it's not the case with the BLI, where the biosensor just dip in the plate containing the samples. So the first impact is that in SPR, we inject the sample inside the equipment. So this sample cannot be recovered for additional experiment. And if the sample are contained in a dirty sample, crude sample, it's not really adapted as you contaminate your equipment. With BLI, the sample is not injected, so it's possible to recover the sample to do additional experiment, but also the crude sample can be used with the BLI. A second point is the time for the experiment. SPR is time consuming because you need to inject a first concentration of your product. After the association and dissociation, you need a regeneration step, and after inject the second concentration and so on. So at each cycle, you can only analyze one sample because you have only one sensor chip. With BLI, biosensors are analyzed in parallel. So we can analyze 8 different biosensors in parallel. So this is, of course, faster, and we don't need regeneration cycle as these biosensors are mainly used as single-use biosensors. So it's faster with BLI, and it allows higher throughput. There is also a point about the evaporation of the sample. So in SPR and BLI, most of the time we are doing the experiment between 25 and 35 degrees. Samples are present in plate with a very low volume. Most of the time it's between 100 and 200 microliters. So of course there is a risk of evaporation. This is mainly the case for the BLI, as there is no cover in the plate because the biosensor need to dip in the plate. So there is no cover and high risk of evaporation. For SPR, it's not really the case, as the plate is covered by a foil and the needle of the biocore is able to pierce the foil to inject the samples. The presence of tag on the molecules is also important. So most of the case, to immobilize your ligand, you need a tag on your molecules. So it's biotin tag or histidine tag. It's really crucial for the BLI. It's a little bit less crucial for SPR, as amine coupling chemistry can be also performed to directly immobilize the molecule to the sensor chip. In terms of affinity, both technologies are able to measure KD from picomolar to millimolar. And in terms of precision, most of the case, SPR show a higher precision. So now let's start with the case studies. So the goal of this project was to develop different methods to characterize the affinity between the TCR and the MHC complex. So we use SPR and BLI technology with several designs. And in terms of reagent, we use MHC and TCR from Immudex. So for the MHC, we use a monomer MHC, which corresponds to HLA-A2 with a specific peptide sequence. This MHC was tagged with a histidine tag. We also use irrelevant MHC for the specificity study, which is exactly the same MHC, but with another peptide sequence. And finally, the TCR was a TCR targeting NY-ESO I with a biotin tag. And it was a modified TCR with an enhanced affinity with an expected KD around 1 nanomolar. So the first design was with the SPR, and we tried to capture the MHC complex. So here we use a CM5 sensor chip, which are the classical sensor chip in Biocore with dextran matrix. This sensor chip was first immobilized with anti-histidine antibodies at high density, around 8,000 resonance units, in order to completely saturate the sensor chip with the antibody. After, at each cycle, we start with a capture of the HISTAC MHC, followed by the injection of the TCR at several concentrations. So for all the SPR, but it's also the case for BLI experiment, we need to inject concentration between 0.1 and 10 times the KD. So here we inject between 0.1 and 10 nanomolar of TCR. After each cycle, we need to regenerate the surface, and we use for this glycine pH 2.5. So here are the results you obtain with this first design. So here in the left is the capture profile. So we start to inject the MHC complex to immobilize the MHC complex to the sensor chip. So we can observe an increase of the signal, but when we stop the injection, we directly observe a decrease of the signal, meaning that there is an instability with the MHC complex, and this MHC complex probably detached to the surface when we stop the injection. So this is probably due to the fact that MHC is a heterodimer, which is not really stable, and we can lose the beta-2 microglobulin part of the molecules when it's immobilized. If we look at the second part of this graph, this is the signal obtained when we inject the TCR. We can see that the signal is very low, around 4 or 5 resonance units. And this is probably due to the fact that the tag present in the MHC is present in the beta-2 microglobulin part. So probably that most of the MHC immobilized on the sensor chip were not active and able to bind to the TCR, and that explains the low level of signal. Now, if we look at the 1:1 fitting, which is applied to the data, the 1:1 fitting is correct, and we obtain KD around 1 nanomolar. So we try to reverse the system and directly immobilize the TCR to the CM5 sensor chip. So here, the TCR is bind to the sensor chip using amine coupling chemistry. So the lysine residue present on the TCR can bind to the dextran matrix of the CM5. It's important to note that with this design, we cannot control the orientation of the TCR. So if the lysine is present on this part, it's perfect because the TCR is well oriented and can interact with the MHC complex. Now, if the lysine residue are present near the binding site, of course, the TCR is misoriented and we won't be able to interact with the MHC complex. So for this reason, we try to immobilize at high density the TCR, so around 2000 resonance units, and after inject the MHC complex between 0.1 and 10 nanomolar. Of course, between each cycle, we have the regeneration step. And if we look on the result, we obtain this sensorgram, which is quite nice, with a perfect 1:1 fitting, a nice association and dissociation phase. If we look at the constant, so the kinetics constant, we obtain around 10 to the 5 for the association rate constant and 10 to the -4 for the dissociation constant. And in terms of affinity, the KD is 1.1 nanomolar, which was clearly expected. If we look at the absolute signal here, we can see that we obtain around 60 resonance units for an immobilization of 2000 resonance units. So the signal is low compared to the immobilization level. And this proves the misorientation of the TCR when it's immobilized. And if we look at the sequence of the TCR tested, we observe that most of the lysine residues were present near the TCR binding site. To overcome this issue, which is not really an issue because the data we obtain are quite okay here, we just need to immobilize a higher level of the TCR. So we perform a third design for SPR where we use we immobilize the bioattenuated TCR on streptavidin sensor chips. So here, the sensor chips are already coated with streptavidin, can bind the bioattenuated TCR. As we can control the orientation of the TCR, we decrease the level of immobilization. So here we target 100 resonance units of immobilization. And after the immobilization, we start with the injection of the MHC complex. Here are the data we obtained. So we obtain this kind of sensorgrams. So as we can observe here, the fitting is not very perfect, mainly for the high concentration. and the signal is quite low. So we obtain between 10 and 12 resonance units for 100 resonance units of immobilization. So there is no clear reason why this sensorgram is not perfect as the design 2, but probably is due to the fact that we work in a fluidics environment, and as the MHC is not very stable, it could partially dissociate in the fluidics and cause this low signal. Nevertheless, if we look at the kinetics data, we obtain quite similar result compared to the design 2, with 10 to the 5 for the Ka, 10 to the minus 4 for the Kd, and Kd between 0.6 and 0.7 nanomolar. The next design was BLI. So in BLI, there is no fluidics, so maybe less interference with the MHC complex. So the first assay we tried to develop was the use of histidine biosensors, which are biosensor coated with anti-histidine antibody. We try first to capture the his-tag MHC and inject the bioattenuated TCR. During the first assay, we had a very surprising result because we had a lot of non-specific binding when the TCR was injected. So we tried to do some investigation and some discussion with the suppliers of biosensors, and it appears that actually histidine biosensors are not really histidine biosensors. They are streptavidin biosensor on which the supplier already coated a bioattenuated anti-histidine antibody. So of course, during this first run, the bioattenuated TCR was able to bind to the streptavidin free site on the biosensors. So the solution was to add a blocking step with the biocytin, which is an analog of biotin. And this small molecule is able to bind to the free site, streptavidin free site, and in this case, avoid the non-specific binding of TCR to the biosensors. So here are the results. So in the left, it's the full sensorgram. So with the first baseline, the loading step. So here we tested 7 different concentration of the MHC complex plus 1 blank, just to see if there is non-specific binding. We have new baseline and the biocytin treatment. And after here, you can see the association with the TCR and the dissociation. So this first experiment was performed in order to determine the optimal MHC concentration for the loading. So here the goal is to have a linear increase of the signal during this first step. So we try to avoid this kind of curves where the saturation is reached very fast and can give some heterogeneity in the binding. And the second parameter is the total binding we observe with the TCR. So we try to obtain value around 0.5 nanometer of response when the TCR is added. So based on this, we selected the purple curve here, which correspond to the 0.25 microgram per ml of MHC complex. And this gave a value around 0.6 nanometer of signal. The second objective of this test was also to control the biocytin treatment. So here we can observe in the blank biosensors, here in red, that there is no signal when the biosensors is loaded, because of course there is no MHC complex. We have the biocytin treatment, and after we inject the TCR, and we can see here there is no signal when the TCR is injected, meaning that the biocytin treatment is efficient. If we look at the sensorgram, we can see it a first step of association, which is quite similar what was obtained with the SPR experiment, with a Ka around 10 to the 5, so it's quite similar. But if we look at the dissociation, we can see that the dissociation doesn't occur in this condition. And this leads to a very high affinity with Kd around 10 picomolar. So this is quite strange. And the main hypothesis is the fact that when the MHC is bind to the biosensors, it can cause some modification in the conformation of the molecules. So we decide to test a last design, which is the binding of the TCR to the biosensor. So here we use streptavidin biosensor to catch the biotinated TCR, and we analyze after different concentration of the ISTAC-MHC. And with this design, it works quite well. So if we look at the sensorgram here, we have a nice association and dissociation phase with a perfect 1-1 fitting. And if we look on the kinetics data, we obtain also 10 to the 5 for the Ka, 10 to the -4 for the Kd. So exactly similar to what was obtained for SPR, and with a Kd around 0.8 nanomolar, which was expected. So we decide to test a little bit more this assay and to qualify this assay. So we decide to test the precision of the method and the specificity. So the precision was evaluated during 3 different runs. These runs were performed by different analysts on different days. And during each run, we prepare 3 independent preparations. So here in this table, you can see the value of the different constant. So for the association, the dissociation, and the affinity constant. And if we look on the CV, percentage CV, so for the repeatability, which is the intra-assay precision, we obtain percentage CV between 10 and 15%. And for the intermediate precision, which is the inter-assay precision, we obtain CV between 10 and 20%, which is quite usual for BLI. For the KD, we have a value around 0.7 nanomolar, so it's quite expected. And finally, about the specificity, we have tested 2 different MHC complex. So the first one is the relevant one with the correct peptide sequence, and we obtain very nice sensorgram. And the second one is the irrelevant MHC with another peptide sequence. And as we can observe here, there is no signal at all with this MHC, proving the specificity of the method. So now it's time for the conclusion. So the interaction between the TCR and the MHC is, of course, very crucial for the T-cell mediated immune response. It's possible to modify this affinity, and when we modify this affinity, this can have significant impact for the specificity, the cross-reactivity, the response strength, and the effector lifespan. So different companies try to modify this to modify the affinity and to also modify the efficacy of the treatment. The affinity can be measured by different technology, including SPR and BLI. And during this project, we showed that we were able to develop different kind of assay with this technology and to demonstrate a KD around 1 nanomolar as expected. Now, if we have to choose one technology, I will go with the BLI, as this technology shows several advantages compared to the SPR. So mainly the speed of the analysis, the high throughput. It's possible to recover the sample, and as there is no fluidic with BLI, this has less impact on the instability of the MHC complex, and this gives better fitting with the 1-1 model. There are also different points that are really important to consider when we develop this kind of method. So the first point is the choice of the immobilized molecules. So of course, we can immobilize TCR or MHC, but we have seen that MHC is not really stable. And when we immobilize the MHC on the biosensor or in the sensor chip, we have sometimes some strange results, some heterogeneous binding. The orientation is also very important, because if your, for example, your TCR contain lysine residues near the binding site, you immobilize this TCR in the wrong orientation, and this can lead weak signal and, of course, non-reliable data. The presence of tag is important for this kind of experiment, but of course, the tag must be different between the TCR and the MHC. because if it's the same tag, you will observe non-specific binding. And this presence of tag is, of course, can impact the run design. Finally, the last but not least, the expected affinity is also very important because this affinity drives the concentration to be tested. So in SPR or BLI, you need to analyze concentration between 0.1 and 10 times the KD. So in our case, we work with a nanomolar range TCR, so it was quite easy because we use concentration between 0.1 and 10 nanomolar for the TCR or for the MHC. So these concentrations are quite easy to produce. They are not so expensive. It is expensive for the TCR, but still quite okay with this amount of product. So just for information, for the whole study, we ordered 20 microgram of TCR and 20 microgram of MHC complex, so it's still quite okay. But of course, if your TCR has a micromolar range affinity, it's more complicated because you need 1,000 times more of the product. It's of course complicated because the cost is very expensive. But the second point is also a constraint for the manufacturing because you will need concentration of TCR or MHC between 10 micromolar. So this is the concentration in your well. But of course, if you want to avoid buffer effects, your stock concentration must be 10 or 100 times higher. So you will need to deal with concentration around millimolar of TCR or MHC, which are expensive, but also complicated to produce due to formulation issue, to protein concentration. So it's interesting to test also this low affinity TCR, but it's really a challenge to obtain the correct reagent. So finally, to finish this webinar, 2 additional information. So following this collaboration with Immudex, we wrote an application note that summarizes all the data that I have presented today about the affinity, about SPR, BLI, and the different protocol we use. This application note is already present in the Quality Assistance website and can be downloaded using this QR code. And finally, we have also a collaboration now with Immudex. So Immudex is able to produce functional TCR and MHC with very high quality reagent with good QC. And following this, Quality Assistance is able now to determine the affinity of the TCR and MHC complex. So thank you for your attention, and we are now ready to respond to the different questions.